Hey, so let's finish talking about the different requirements that we need to be careful of when we're growing and culturing bacteria. So remember to review that there are four different characteristics that we need to control for. First one was temperature. Those are the ones that we already talked about. Then there is oxygen requirements, and then there is pH as well as water availability. So we already looked at the temperature one, so now let's move on in our notes and check out the oxygen requirements. So based on oxygen requirements, the prokaryotic organisms can be divided up into 5 different groups. There are those that are considered obligate aerobes, and I want you to think about what the word obligate actually means, right? That word right there. If something is obligated to do something, they have to do it, right? An obligate aerobe has an absolute requirement for oxygen to generate their energy because they use aerobic respiration. So one example is the genus Pseudomonas. Another category is called the obligate anaerobes. So this means that these organisms absolutely cannot multiply if oxygen is present and are often killed because their cells end up making toxic oxygen derivatives. So bacteroides that are present in your large intestine, which is an entirely anaerobic environment, as well as Clostridium botulinum that causes botulism, are 2 examples of obligate anaerobes. Facultative anaerobes grow better if oxygen is present, but can survive in the absence of oxygen too. So I like to think of facultative as they have the faculties to be able to grow with oxygen. They have the faculties to be able to survive without. So a facultative anaerobe uses aerobic respiration if oxygen is there, but it can also use fermentation or anaerobic respiration in the absence of oxygen. So the example here is E. coli. Now, group 4 would be the microaerophiles. The microaerophiles are the ones that require very, very small amounts of oxygen, only about 2 to 10% in the solution that they're in. So, and they use it for aerobic respiration. If there's higher concentrations of oxygen, it's actually inhibitory and they won't grow very well at all. So one example is Helicobacter pylori. Helicobacter pylori is the bacteria that they have discovered causes ulcers in your stomach. So you can see what we're talking about here. You sometimes get air into your stomach. The bacteria can survive small amounts of small amounts of oxygen being in there, but not very much. Then there are the aerotolerant anaerobes. And these would be organisms that can exist in oxygen, but actually don't use it for energy. They're quite indifferent to oxygen as they don't use aerobic and anaerobic respiration. They're also called obligate fermenters. Streptococcus pyogenes is actually what causes strep throat, is actually a really good example of an aerotolerant anaerobe. Okay, it's anaerobic, but yet it's in the back of your throat. So it's being exposed to oxygen constantly, but it doesn't kill it. Now, this next part, I want you to really take a look and focus on the different types of oxygen requirements and where we might actually find the bacteria growing in a culture. So with the example here that's down at the bottom of the page, okay, what I want you to take a look at is look at where the red colonies are growing. These tubes have broth in them. So if I put bacteria into a broth and allow it to grow overnight, where would I find the bacteria based upon their oxygen requirements? So check it out. The obligate aerobe grows up here at the top. And you can see that there are actually some pretty large colonies up there. Now, the obligate anaerobe, hopefully you would expect to grow down towards the bottom of the tube. Not as much oxygen down there, right? As a matter of fact, there really shouldn't be any at all. Whereas now we've got the other three. I think the microaerophile is the easiest one to figure out because it requires 2 to 10% of oxygen. So that means it's going to grow towards the top of the tube, just not up at the very top. So that makes sense that it's growing right here. Now the hard part is taking a look at the difference between the aerotolerant tube and the facultative anaerobe. What do you notice is the difference? I hope that you're recognizing that with the facultative anaerobe, that the colonies that they're showing growing in this tube are actually much larger at the top than at the bottom. So remember, the facultative anaerobe grows better in the presence of oxygen, but can still survive without it. Whereas the aerotolerant doesn't matter. They are entirely indifferent to the presence of oxygen, so they don't grow better at the top. They don't grow better in the bottom. They grow anywhere in the solution. So what I'd like to do is then look at the reasons why. Why is it that oxygen can become so toxic to some microorganisms? Oxygen itself is not toxic, but it can be converted into a number of compounds during metabolic processes that are highly toxic, such as superoxide and hydrogen peroxide. I'm guessing one of those just rang a little bell in your head. And I want you to think back. What do you know hydrogen peroxide from? Do you remember? Do you remember when you were little and you scraped your knee and maybe you went inside and your mom kind of washed it off? She probably put some hydrogen peroxide on it. You maybe remember it burning a little bit, and at the same time, lots and lots of bubbles formed. That's actually a really good thing. So keep that in mind, and now let's take a look at this next bullet. In order to survive in the presence of oxygen, cells must have enzymes to degrade toxic products into non-toxic forms. So catalase breaks down hydrogen peroxide into water and oxygen. Look at that. Water and oxygen, a gas, creating bubbles. Get it? Superoxide dismutase is the enzyme that degrades superoxide to produce hydrogen peroxide. Interesting. But if you've got superoxide dismutase and it degrades superoxide into hydrogen peroxide, isn't hydrogen peroxide even toxic? Yep. So that means that bacteria would have to have both. Check this out. Look down here at the bottom of our previous screen, right? The obligate aerobes have superoxide dismutase and catalase, whereas a facultative anaerobe has catalase and superoxide dismutase. An obligate anaerobe doesn't have either. It doesn't have either of those types of enzymes. The microaerophile has small amounts of catalase and superoxide dismutase, whereas the aerotolerant only have the superoxide dismutase, which is why they can't grow super great like the facultative anaerobe in the presence of oxygen. So that's why oxygen can be toxic to some microbes. Now let's check out pH. So our normal internal pH is 7, or a little bit higher. Sometimes it's between 7 and 7.4. So bacterial cells, which grow within the range of pH between like 5 and 8, is where they can normally grow. And those that have a pH that's optimum near neutral are called neutrophiles. Remember, we talked about that suffix phile in our previous lecture. This gets exploited by phagocytes to kill ingested bacteria in acidic endosomes. So those lysosomes on the inside of the white blood cells actually have acid in them, so that way they can destroy the bacteria a little bit more successfully. Now, some bacteria may adapt to grow at low pH. Again, Helicobacter pylori grows in the stomach and causes ulcers, so obviously it can survive closer at around 5.5, it's known as an acidophile, whereas those that grow optimally at a pH above 8.5, those are called alkalophiles. When we look at water availability, all cells, everything on Earth requires water for growth. It's how they manage the influx of water into their cell, or the amount of water that's even available to them that's important. So the local concentration of dissolved ions, like salt and sugars, can affect the osmotic balance of a cell. This is what you studied in biology, right? It requires active transport of ions across the plasma membrane to maintain cellular structure. So if the concentration of solutes is low outside the cell, water will tend to flow inside the cell. If the concentrates of solutes is high outside the cell, water tends to flow out of the cell due to osmosis. This causes the cytoplasm to dehydrate and shrink from the cell wall, a phenomenon called plasmolysis. Bacteria that can tolerate high salt concentrations, up to nearly 10% salt, are called osmotolerant. Staphylococcus species, which are present on the dry environment of the skin, are an example of osmotolerant. Now, if we move on and take a look at nutritional factors, I had referred earlier in our previous lecture about how the agar that we use, that there's different types, and we can add different types of nutrients to different types of agar. So in order to synthesize all cellular components, a variety of nutrients are required. Sorry, had to have a drink of water. Prokaryotes only have the remarkable ability to use atmospheric nitrogen as a nitrogen source. So they're very lucky. We can't. The major elements are the elements that would make up the cell constituents and include carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, potassium, magnesium, calcium, and iron. Okay? The big ones are CHOPNs, CHOPNs, CHOPNs. That's a little acronym that you can remember. The trace elements are required in very minute amounts by all cells, and they include cobalt, zinc, copper, molybdenum, and manganese. A lot of those elements are required for enzyme functions, okay, like the superoxide dismutase and catalase. So having some of those trace elements is going to be super important when we ask those bacteria to grow in culture for us. Now, when we look at carbon sources, you've studied this as well. You looked at this in biology. So the source of carbon distinguishes different groups of prokaryotes. So prokaryotes that use organic carbon are called heterotrophs, right? Kind of like you. You require organic carbon. You got to take it in. You got to eat it. Medically important bacteria typically use organic carbon in the form of glucose. Prokaryotes that use inorganic carbon in the form of CO2 are called autotrophs. They're very important in carbon fixation, so part of the carbon cycle. So when we look at the energy sources that we might see between microbes or between bacteria is going to be that of either sunlight for those phototrophs. So plants, algae, and photosynthetic bacteria are considered phototrophs. And then organisms that obtain energy by metabolizing chemical compounds are called chemotrophs, like mammalian cells, fungus, and a lot of bacteria. So this is a description of different types of media and the characteristics that are used to be able to create that agar and then determine whether it is helpful for us to use for the type of bacteria that we're trying to isolate. So there's complex media where there's a lot of peptones and extracts. The media that we've used so far has been complex media, okay? We've also used a little bit of nutrient agar. It usually supports the growth of a lot of different bacteria. So when we were doing our ubiquity lab, that was the kind. When we looked at the streak plate method on our previous lecture, we looked at blood agar as an example, and I talked a little bit about how it's typically used in clinical labs. So the idea here is that there are different types of media that are used to cultivate bacteria. Some of them are selective and some of them are differential. What do you suppose they're used for? Well, this is where I want you to go back and I want you to think about differential staining. The whole point of differential staining was to identify the differences between two different groups. So differential agar is going to end up doing the same thing. Selective means that you're really trying to focus on getting just one species. It is going to help you select for a specific type of bacteria, and only that bacteria would be able to grow. So let's continue taking a look at this and check this out. So differential media. Blood agar is used to detect bacteria that produce a hemolysin. So the type of hemolysis is an identifying characteristic. So when you look at example A and the picture, you can see that there's a colony, and then going around the outside, there's kind of a clear ring going around the outside of it where all the blood in the agar has been hemolyzed. And so that's an example of what Streptococcus pyogenes does that causes strep throat. It makes that clear zone. So it makes it really easy for us to visually identify that type of bacteria. So when you have to have that strep test done, that's what they're swabbing for. In contrast, species of Streptococcus that reside in the throat harmlessly cause what's known as alpha hemolysis. So that's the picture on the right labeled B. It's characterized by a zone of like a greenish clearing. So you can see the green on that one. It's pretty obvious. Now, we kind of got some stuff cut off over here because of course I forgot. I used Google Slides. But MacConkey agar is bacteria that ferments and produces an acid, which turns the pH indicator a different color. So the example here in this picture on the lower right left-hand corner, sorry, that's covering up half of my words, basically is E. coli forms pink colonies because it's lactose fermenting, so it's producing the acid. Whereas those that are lactose negative bacteria will form tan or kind of a whitish color, like a cream-colored colony. They don't turn pink because there is no acid being produced. They're not a fermenter. All right. So, stuff is cut off again. This is learned. All right, so providing the right atmospheric conditions. So when we're trying to study obligate aerobes, the broth cultures do better when you take the tubes and you kind of shake them or finger vortex them. We talked about that a little bit after our first lab, taking the tube and tapping the finger across the bottom of it. If you recall, kind of looks like doing this type of thing to the bottom of the tube, okay? Then if there's increased carbon dioxide, the growth of many medically important bacteria can be enhanced by providing that increased amount of CO2. So studying Neisseria and Haemophilus will do that. Obligate anaerobes, boy, theirs is a challenge, right? We need oxygen. So how in the heck do we study something that's anaerobic that can't be exposed to oxygen? Well, you can see down here in this anaerobic chamber that there are these, boy, how do I describe those? They're cylinders, and then they've got a screw lid, and then they have a vacuum attachment up here at the top. That enables you to use a vacuum to extract the air out of that cylinder. So then you can transport it and put it in the chamber, and it stays in an anaerobic environment. So that's an example of an anaerobic chamber, of an anaerobic jar, and there are other reducing agents as well to be able to study anaerobic organisms. Now, the next thing we want to talk about, I believe, is where I'm supposed to stop. Yep, I'm going to stop right here on enrichment cultures. So I hope you guys made it through the lecture okay and you didn't fall asleep on me. So you're going to be taking a look at something else for the remainder of the period today. If you've got all your notes done up to this point, nice job. I'll see you when I return.